databases, aiding future diagnostic development and standardization. I'd like to share a very interesting example here. Back during the Omicron outbreak, WHO came out with a regulation where they said for countries using diagnostic kits that include S-gene target failure or SNP detection assays to screen the variants, those samples should be recommended for sequencing and to characterize the circulating SARS-CoV-2 variants. And they said whenever available, these genomic sequences and the S-gene target failure data should be shared with the WHO. Now what we did during this period, we sequenced all the samples that were available and to check if these S-gene target failures were actually Omicron variants. We saw a very interesting graph. The blue ones show the blue lines show the isolates which showed S-gene target failure, and the orange ones which were non-S-gene target failure samples. So you see a very nice wave there. Towards the end, as we know, the Omicron, towards the end of December, the Omicron cases were rising. So what we saw, so what should we expect? That the S-gene target failure should be increasing. But what we saw was completely opposite. S-gene target failure cases started decreasing. So did that mean that Omicron cases started falling? No. It was the Omicron had evolved into multiple lineages, that is BA.1 and BA.2. BA.1 showed S-gene target failure, whereas BA.2 did not show S-gene target failure. And here we missed detecting BA.2 if you would have used that particular kit. So here it guides us whether to continue to use a particular kit or to discontinue it. So it helps in policy making. Now with the ever-evolving nature of pathogens, traditional method often falls short to address the emerging challenges. So it offers a groundbreaking approach to revolutionize how we design our vaccines. So vaccines have always been a cornerstone of public health, but methods used to develop them have evolved significantly over time. Now, NGS has allowed the researchers to predict the antigenic proteins exposed on the surface of the pathogen, and using reverse vaccinology approach, these predicted antigens can then be experimentally tested for their ability to elicit immune response. The most remarkable outcomes of NGS-driven vaccine development is the rapid creation of mRNA vaccines for SARS-CoV-2. Now, why this matters is with NGS, we can address the challenges like antigenic variability and emerging pathogens in real time. This is especially critical in responding to outbreaks where rapid vaccine development is essential. It also enables the design of vaccines tailored to specific strains circulating in different regions, increasing their effectiveness. Now again, I would like to share a study from IISc Bangalore, where the study highlights the significant mismatch between currently available dengue vaccines and the dengue strains circulating in India. It shows antigenic divergence where the prevalent Indian dengue virus strains differ significantly from those used in the tetravalent vaccines, which are based on the older strains isolated from 1964 and 1988. Following this study, researchers developed a novel DNA vaccine candidate combining the envelope protein domain 3 of dengue virus serotype 1 to 4 and non-structural protein 1 of dengue virus 2. Now the study emphasizes the utility of the consensus genus approach in conjunction with DNA delivery for the development of vaccines against various emerging and re-emerging viruses. Now coming to another transformative application of genomics, that is leveraging the human microbiome for therapeutic development. The technology has revolutionized our understanding of human microbiome, an intricate community of bacteria, viruses, fungi, and archaea residing in and on our bodies. The microbiome is now recognized as a key player in health and disease. We know very common examples like recurrent Clostridium difficile infection, where fecal microbiome transplant is already in use, and there are several clinical studies which are undergoing to investigate the use of vaginal microbiome transplantation to treat symptomatic and intractable bacterial vaginosis and in HPV infected women. Another critical and the last application of NGS that I am going to discuss is the transformative impact on clinical practice, precision medicine, particularly in oncology. It allows the discovery of tumor-specific mutations, enabling personalized treatment, plans tailored to genetic profile of an individual's cancer. By detecting mutations associated with hereditary cancer syndromes, NGS supports early risk assessment and preventive strategies. It also aids in selecting targeted therapies by identifying actionable mutations. Now, this is a very powerful example of a next-generation sequencing which is transforming the clinical practice in real time. Using NGS combined with machine learning, surgeons could achieve enhanced diagnostic accuracy for central nervous system tumors even during surgery. So, within 90 minutes during a live surgery, they could take a decision whether to resect a brain tumor or not to resect a brain tumor. So that is the real-time application of NGS where surgeons could take a decision within 90 minutes to treat a brain cancer. Now another example, and the last example, perhaps which illustrates how genomics can play a life-saving role in chemotherapy safety and precision medicine. Certain drugs such as fluorouracil and capecitabine can cause severe and even fatal toxicity in patients carrying genetic variants in the DPYD gene. Now the statistics are striking. 2 to 3% of patients carrying one DPYD variant can experience fatal toxicity. Now this highlights the critical need for pre-treatment DYPD genetic testing to identify at-risk individuals. Now genomics not only helps prevent adverse outcomes but also ensures the patients receive safer and more personalized therapies, revolutionizing the cancer cure. To summarize, genomics is revolutionizing healthcare by transforming diagnostics, enabling rapid and accurate pathogen identification while guiding pandemic preparedness through tools like wastewater surveillance to track emerging variants and zoonotic diseases. It has redefined vaccine development, addressing antigenic diversity to create region-specific and effective vaccines and advanced precision medicine by tailoring treatments to individual genetic profiles for improved outcomes. Together, these advancements highlight the immense potential of genomics to tackle global health challenges and improve patient health care. Thank you.
Thank you, Dr. Rashmita, for precisely telling us the role of genomics in various diagnostic and research fields of microbiology. Thank you so much. It gives us a proud feeling when a new generation is delivering a talk is our own student. I call upon Dr. Madhuri Somani. Madhuri Somani is a University Gold Medalist from BJ Medical College, Pune. She is currently working as Consultant Microbiologist and Infection Control Officer at Pushpavati Singhania Hospital and Research Institute at New Delhi. She is the Secretary of Hospital Infection Control Committee and the ANSP. Dr. Madhuri has recognized for proficiency in leading and managing the high-quality microbiology laboratories, particularly in tuberculosis diagnostic. She has played a vital role in implementation and maintenance of hospital infection control programs credited for zero non-compliances. She is a faculty member for Outbreak Investigation Workshop, sharing knowledge and skills in managing infectious diseases outbreak effectively. She has authored multiple scientific papers and contributed to both chapters on diagnostic microbiology, infection control, and biomedical research. She is a mentor and guide for clinical DNB residents, fostering the next generation of healthcare professionals. I request Dr. Madhuri to deliver a talk.
Good morning. Thank you so much, ma'am, for that kind introduction, and I take this opportunity to express my gratitude to the organizers to give me an opportunity to speak today. So with whatever little time is left, now I'll try my best to stick to the time schedule. Keeping in sync with the futuristic theme of this conference, I shall be speaking upon total lab automation the road ahead. So I'll be delving in the next 15 odd minutes on the why and what of total lab automation, the initial impediments and drivers towards total lab automation, what are the current systems available and their benefits, and with them come challenges and future directions as well. So, in a technical definition, what exactly is automation? Automation is essentially a technology by a procedure that is performed without human assistance and intervention, and in contextual laboratory, it's essentially switching from manual, repetitive, moderate value tasks to machine-based tasks. And I think our clinical biochemistry and hematology counterparts are already doing their entire workload on the 6-mexes and abbots, and about 80 to 90% of their work is already automated. So automation is relatively late to hit our field. It's only been 30 years since the first automation systems, starting with the blood culture systems, actually came into the market. Following that came the ideal susceptibility testing equipments as well. And those of us who have been using Bactex, Bacteroids, Vitex and Phoenixes know how well this partial automation has actually changed the way we work and how much it has contributed to the productivity of a clinical microbiologist's work. So if partial automation can add so much value, just imagine what total lab automation could do. So let me just be clear about this, that what we're talking about here is the automation of the steps of the clinical bacteriology diagnostic workflow that would start from inoculation to the final reporting. And this particular entity, TLA, has only recently been recognized as a valuable